We can dive right in. I'm Robert Abel, CSO of Schrödinger. And our work is to accelerate drug discovery by way of deploying ultra-large-scale deployment of advanced predictive modeling. So it's important to acknowledge at the outset of a presentation like this, discovering a drug is really, really hard. It should be of no surprise to most of the attendees. And in order to actually discover a drug, you have to find some molecule that simultaneously optimizes all of these different properties. And as was shown earlier in the morning, you might find a potent molecule pretty quickly, as was shown for CDK20, but to have that molecule be positioned to enter into clinical trials, in addition to potency, you have to optimize all these other properties necessary for that ligand to become a drug. And that's a major contributor to, in our best estimation, about 66% of industrial drug discovery projects ultimately failing to advance a drug therapy into the clinic. The animating vision for Schrödinger as a company is to eventually develop computational methods of sufficient power that will be able to evaluate some approximation to the universe of all synthesizable molecules that might be of interest to the drug discovery project, and then pluck from that single distribution through application of these predictive techniques, the single best molecule that best optimizes all of these different properties simultaneously, such that it can be advanced with confidence into animal studies and ultimately the clinic. We're far from that reality right now, but enormous strides have been made that I'll be detailing in what follows. So historically, there's been two major approaches to this problem of how do you predict the right molecule for your drug discovery project. There's been data-driven methods, and these are most typically referred to as machine learning and artificial intelligence methods. These methods are very, very good in interpolation. They're enormously computationally efficient. They can handle and often require very large training sets. But the real knock against these methods is they often cannot extrapolate to the novel chemical spaces of greatest importance to drug discovery. At the far other extreme are physics-based methods. These methods are interesting in that they don't, in principle, require any sort of training set, and if you deeply understand the property you're predicting, they can extrapolate into novel chemical spaces with high accuracy, but they're extremely computationally demanding and can be quite slow to apply in active drug discovery projects. So where Schrödinger has really been breaking new ground in our view is developing these integrated methods that benefit both from physics-based techniques and machine learning methods such that we can develop computational tools that are fast, accurate, can handle very large data sets, but can also extrapolate into the novel chemical spaces of greatest interest to drug discovery. So I wanted to give a little bit of a visual representation of how this works. This is an example of a free energy calculation in action. The purple is the protein, the yellow is the ligand, and we want to further optimize this ligand to be a small molecule therapeutic. We can have a design hypothesis, for instance, introducing an aryl halide, and if we keep track of the fluctuations of the energy as the system is wiggling and jiggling with respect to temperature, we can work out a very precise and accurate prediction of the binding free energy of that molecule. That's a critical parameter to optimize if you're looking to have an efficacious small molecule to advance into the clinic. So the sole justification of running that very expensive simulation is the accuracy of the prediction. We've done a number of large-scale benchmarking of the accuracy of this technique, and we find that it actually approaches the inherent experimental error of the target data. So if you just measure a lot of these molecules twice, the amount of error you get in the experimental readout is comparable for many cases to the error that you're getting for the predicted affinity of that molecule. So that's very exciting because that means we have the equivalent of a computational assay. So it's approaching the experimental error for amenable targets, but it's 50 times faster per molecule to evaluate these molecules using free energy calculations computationally, where we can do that in something like 6 hours of wall clock time, where it takes about 3 weeks of painful experimental work to interrogate those molecules similarly in a wet lab. The throughput is 250 times greater, where a fully staffed medicinal chemistry team will typically synthesize 5,000 intentionally designed molecules over a 5-year period. We're utilizing these types of computational techniques. We can evaluate that same set of molecules in about a week using cloud resources. That allows the discovery project to focus their synthesis resources where they will be most productive. So that's if you want to evaluate thousands, but the chemical space available to our project teams is vast. You can see these crazy estimates that may be as high as like 10 to the 60th. To deal with these very large chemical spaces, that's where we realize tremendous benefits by integrating these physics-based simulations with the power of artificial intelligence and machine learning methods through an active learning strategy, where we can't run free energy calculations on 1 billion molecules, but we can advance subsets, 1,000 randomly selected molecules into these physics-based simulations. And then once we have that training set data, we can then build a machine learning model to that physics-based data, that virtual data, and then you can use that to re-rank the billion and then advance an improved set to additional physics-based simulations. And then you can iterate this approach using an active learning framework until you finally have a good set to advance to synthesis and assay. Based on extensive retrospective and prospective profiling of the accuracy of this technique, we would expect about 8 of these 10 molecules to materially move the drug discovery project toward its intended goals. So this is the basic idea that underlies the entirety of our computational platform, where we now have technologies that can assess the druggability and the tractability of different targets through binding site identification and druggability assessment. Once we have those putative small molecule binding sites, we can find hit molecules that have some potency for that target. And then we can use both experimental inputs in these physics-based simulations to determine binding modes of those hits. And then we can do large-scale ideation in silico, where this is another place where the machine learning and artificial intelligence methods make really important contributions, enumerating the space of ideas that may be interesting for the drug discovery project to interrogate. We can then do multi-parameter optimization in silico using these combined physics and machine learning methods. And if everything works perfectly out of the box, you might think you might have a development candidate molecule coming out of that can go directly into animal studies in the clinic. More commonly, you actually learn things in this experimental step about where the chemical matter needs to further improve. That can be analyzed with machine learning methods, and then we can iterate this flywheel where the details of the ideation, the multi-parameter optimization in silico is being altered and improved based on these learnings that are coming from analysis of the experimental data. And then through iterating this cycle, we can rapidly triangulate some development candidate molecule that may be of sufficient quality to advance into clinical studies. So Schrödinger uses this technology platform to support 3 different lines of business. The first is software licensing to pharmaceutical companies to help them support their work through access to the technologies. We have a collaborations business where we work with biotech companies and pharmaceutical companies to help to advance their projects with them side by side. And most recently, we've started to pursue our own proprietary pipeline of wholly owned assets we're developing into the clinic. And the technology platform really does support all those different activities. So in terms of validating the approach, our collaborations business has led to a number of different therapeutics in different phases of clinical trials, including one product on market, as well as other collaborations that are moving along quite nicely in the same direction. The other thing we've been pursuing is our own wholly owned drug discovery operations, where 3 targets, these are all of oncology interest, very much in line with the goals of this day. They've been advanced into the clinic. They're working their way through phase 1 studies, and time will tell what will come of these. To make it a little more concrete how we use this technology platform to support drug discovery operations, I wanted to present briefly one of these programs. So MALT1, it's a clinically validated oncology target. Once the target is validated, our goal then is to find better chemical matter that will inhibit or activate the particular protein, MALT1 in this case, and try to find something that's suitable to be advanced into clinical studies and hopefully eventually become a marketed therapeutic. Here's how that worked in practice. And to put sort of a sense of scale on the numbers, I'm drawing an analogy with sort of global populations. So in order to find that molecule that was of sufficient quality to advance into the clinic, we had to interrogate 8.2 billion molecules with machine learning methods. These machine learning methods were trained to these 12,000 computationally intensive physics-based simulations. And it was an iterative process where, based on the initial results of interrogating these molecules, we ran additional physics-based simulations, which then allowed us to improve the machine learning models to advance better molecules into these physics-based simulations. That's just going from essentially the equivalent of a world population down to the population of a small town. And then once we're at that population of a small town of most exciting molecules, then we were able to synthesize only 78 molecules from the lead series, 29 molecules project-wide, to eventually identify the single molecule that we wanted to advance into the clinic. And it's that very powerful triage of the chemical matter that the predictive modeling and artificial intelligence methods is supporting. That entire process only took 10 months to have the development candidate molecule in hand, and that's really quite fast by industry standards. So just where this is now, phase 1 studies are in progress, and time will tell whether or not this will materialize as a therapeutic for patients. So just to summarize all of this, what we're hoping to move from is a traditional discovery paradigm where you typically synthesize something like 5,000 molecules over a 4 to 6 year period, and then you end up identifying a drug candidate molecule that will often have some property liabilities where you have to kind of hold your nose and try to advance that chemical matter because it's the best that's been identified so far, to a new paradigm where we can evaluate billions of molecules in silico and only synthesize typically fewer than 1,000. That should be done much more quickly and at lower total cost, and ultimately lead to a drug candidate compound with a better property profile, which will make that therapeutic more likely to succeed in the clinic. So where are we going with this next? Toxicity continues to be a major challenge in drug discovery, and we want to adapt this technology not just to optimize on-target potency, but also to extend the reach of the technology to begin to identify molecules and triage out molecules that are likely to be toxic in the body through interacting with a constellation of all your other proteins that you don't want to inhibit because it'll cause problems. So we've been taking that head on. We're now, in addition to modeling on-target potency, we're building out predictive modeling capabilities for all the other constellations of off-targets that one wants to avoid in a drug discovery project. And we've been delighted that the BMGF has gotten really interested in this to support some of their global health efforts. They've committed $10 million to support the effort. NVIDIA also got really interested in it, and they're helping us to further streamline these technologies to make better GPU computing utilization, as well as working with us in some of the AI/ML sides of this modeling stack. So leveraging those technology enhancements, we're hoping to move from this traditional paradigm where you first optimize on-target function and efficacy and then worry about safety and toxicology much later in the process, to a new paradigm where at the start of our drug discovery projects, we can simultaneously optimize efficacy and safety using these types of in silico approaches. And yeah, I just want to quickly thank everybody spearheading all of the work that's reported here, and also want to thank GCP. Applying modeling at this scale requires access to huge cloud computing resources, and they've been wonderful partners to enable that to happen. So with that, thank you so much. Thanks.
Hi, my name is Jimmy Lin. I'm the Chief Scientific Officer at Freenome. And it's great that you guys heard a lot of the great talks this morning about being able to sort of monitor and be able to understand cancer throughout the whole journey. And there was a lot of talk about early detection, and that's what we do. We think that the best way to beat cancer is not wait until it's already metastasized and really complex to fight. But if you can catch cancer early, it's very curable. Or you could catch it before it even develops into cancer. And that's what we do at Freenome. So at Freenome, we have sort of 3 verticals what we do. Number 1 is there's a population health. We want to figure out what are the patients at higher risk so that they need to be screened for cancer. And that's an effort we've been doing with partners such as Oracle Cerner and others. So that's one area. Number 2 is figuring out the people who actually need the screening. We actually develop different blood tests to be able to help to figure out which patients actually have cancer or have precancerous conditions. And that's going to be the focus of what I'm going to talk about today. And then lastly, we work with biopharma partners. If we detect these cancers early, are there ways that we can involve them in early clinical trials, or can we monitor their the different types of disease so we can really give the right drug to the right patient, and also throughout their treatment journey? But what I'm going to focus about in sort of the short talk I'll do today is just the middle part. How do we catch cancers early, and specifically colorectal cancer? So colorectal cancer is the second deadliest cancer. You can see here, In the US alone, there's going to be 53,000 patients that will die from it, so it's a horrible disease. But the number I want you to focus on is 91%. 91% of these cancers can be treated curable if it caught early. So if we're able to sort of catch all these cancers, colorectal cancer could be a very, very easy to deal with disease. So that's the number, 91%. If we can catch them early, it's actually a very easy to treat disease. But the problem is we have a lot of different modalities to be able to screen. We have colonoscopies. There, you'll be able to sort of actually directly visualize the disease. You have a stool test, but it's often very difficult to be able to have patients to participate. So even though that the American Cancer Society is recommending, at least trying to get to 80%, right now in the US, we're only getting about 59% of patients who need to be screened actually screened. So there's millions of patients who have already ready access to these sort of technologies, but they aren't able to take advantage of it. And there's a big barrier there, whether for existing tests. So what we do at Freenome is how do we lower the threshold of this, right? If it's hard for people to schedule a day off and do a bowel prep to be able to get screened, or they're not very a fan of sort of standing in stool, what if we just do a test through a blood test? As they go into their annual physical checkup, they just have extra blood drawn, 2 tubes drawn, and we can sort of screen for starting with colorectal cancer and later other cancers as part of their regular checkup. And this, we think, would really reduce in terms of the gap and be able to raise the patients are actually being screened. And this is not just sort of a theory, is that there have actually been studies that's been done in terms of patients who are not being screened. 2 out of 3 of them says, you know what, if there was a blood test, we would take it. And this has actually been studied, and then other studies have looked at increasing of adherence of sort of 7.5% or more. So this is going to be a very powerful methodology to really increase the number of people screened for colorectal cancer, so we can catch it early, and these patients can potentially be treated much, much earlier. So we've been working at Freenome for the last sort of 10 plus years in terms of thinking about how we do this. Up front, we created a multi-omics platform, be able to really understand and molecularly characterize this in many different ways. Then, through also partner with sort of with the Google Cloud System, really develop computational models to be able to make sense of all these degrees of freedom and this data to really create a classification model there to be able to then sort of show this its efficacy. In the discovery stage, we threw the kitchen sink at it. Everything of omics that we can think of that could be helpful in terms of characterizing the molecular biology within the blood, whether it's through whole genome sequencing, through the methylome, looking at the proteome, looking at the transcriptome, all the omes you can think of, including autoantibodies as well. And this generates a large amount of data. And the key then is, when you have a lot of features, what are the ways to be able to do it so you don't overfit, and you create models that can be able to be generalizable there. What's so important to this, and I think most people know within AI, is it's sort of garbage in, garbage out. The quality of the data is very, very important. Even though we have all these modalities, how can you get the best quality data to be able to do this? So what we've done is, even in the discovery process, we have a prospectively collected patient cohort in the intended use population. We spent a lot to be able to collect all the blood from the right patients, and all of them actually undergo also colonoscopy. So you have the comparison with the gold standard right away. And with this data set then, we're not sort of collaging data sets from public sources that may be hard to sort of combine. We generate our own data set there and be able to then actually train it sort of based off of it. Then how do we validate it? So we started an FDA trial there a few years back, and then we just read out this year, one of the largest in the screening category there. And then ultimately, this is what we'll be able to see, like is the model actually that is developed, is it actually going to be helpful in patients? So excited to say that this year we read out our PREEMPT CRC cohort. It met all the primary endpoints. We're in the middle of working with the FDA to submit it and hopefully to be able to launch it the year following there. And this is only the beginning, right? So CRC already. We know that it has markers already in the blood. And within our test, we have markers for many other diseases. So as we are running these studies and screening these patients for CRC, we are starting our feedback loop, generating data for all the different cancer types, and then also working with our partners, for example, like Oracle Cerner, be able to leverage some of the EMR data. We think that this creates an amazing engine to be able to really collect the largest data set there. At launch, we're expecting within a few years to be able to get to about 1 million patients a year, and this will be larger than many of the sort of the clinical trials in terms of data generation that would be done through commercial samples. So we like to think about, if we were Tesla, this is our Roadster out there generating data to allow us to be able to create the Model S and then ultimately the Model 3. So this is only the beginning, and we're excited that having this sort of multi-omics platform and be able to first screen for CRC. We're working on lung cancer next already, and that trial is ongoing with PROACT, and ultimately doing one cancer at a time, and then buckets of cancer at a time to be able to catch all the cancers early, so then people will no longer be afraid of the word cancer. Thank you.
Great, so thank you so much to the organizers for a wonderful session today. And I'm going to be the third speaker in today's session, discussing transforming drug development with AI-powered digital pathology. And I'm the co-founder and CEO of PathAI. We're a company focused on using AI-powered pathology to improve patient outcomes. We're based here in Boston. And a lot of people may not think a lot each day about what pathology does or the work that's done in the pathology lab. But there's been huge advances in certain areas, particularly the analysis of blood, next-generation sequencing for molecular testing. But really the core of a lot of diagnostics many of us and our loved ones will have to do throughout their life is a tissue biopsy is taken. So it's not blood, it's not just cells, but it's really analyzing tissues in spatial context. And then the analysis of that tissue under the microscope, in which there can be dozens of slides per surgical operation. Each slide may contain on the order of hundreds of thousands of cells, and those cells could be a mixture of 1 or 2 cancer cells and many, many normal cells. And it's the analysis of those samples that becomes the ground truth and the critical diagnosis for determining subsequent treatment decisions, whether that's in clinical trials and drug development. So pathology analysis is critical for the development of new drugs, but also in clinical practice. If any one of us undergoes a biopsy, it will be the pathology report that then becomes the ground truth to guide subsequent treatment decisions. So leveraging the power of digital and AI to solve this problem can have a huge positive impact on patient outcomes. And this has been our mission at PathAI since our founding in 2016, which was about 3 or 4 years after really the first modern, recent, huge advance in artificial intelligence, which was the advances in deep learning in the early 2010s. At that time, we recognized pathology was going to be an area of healthcare that would be absolutely transformed by this technology because it relies on quantitative, objective, reproducible, and highly scalable analysis of tissue samples. And then with the latest advances in the last 3 or 4 years, which I'll talk about here, we've been able to accelerate tremendously the power of this technology to cover a much, much broader set of therapeutic areas and tissue types, as well as outputs than we ever thought would be possible this fast when we founded the company in 2016. A few of the recent highlights I'll get into. In 2022, so just about 2 years now, we released our first publication on our new PathExplore product, which is our way to convert H&E images from essentially every different cancer type into quantitative features that then can drive downstream decision-making, particularly in drug development. But we've always been equally focused on both drug development on the one hand and clinical medicine on the other, and we really see AI-powered digital pathology as a key technology to link the two. And one of our first major advances on the clinical side was receiving FDA 510(k) clearance and CE mark for our AI site diagnostic platform, which our vision is every lab around the world will have access to both digital pathology as well as a platform like AI site to enable them to sign out cases digitally, as well as increasingly to use AI to improve diagnoses and make diagnoses more quickly. In 2023, we launched our AI site product, which we're now deploying to labs globally, which is our image management system for pathology labs. And I'm going to highlight two big partnerships that we've signed so far in 2024. One on the drug development side, really at the center of late-stage drug development and personalized medicine, and the second with the largest diagnostic lab in the US. So the one on drug development is our partnership with Roche Diagnostics, who's a leader in tissue-based companion diagnostics. So these are the new tests that are really at the core of precision medicine, where every new therapy will have a companion that then identifies the right patients for those therapies. And therapies that fall into that category need a companion diagnostic. We and our partners at Roche believe AI and digital pathology are going to play an important role in the future of companion diagnostics. And the second is this partnership with Quest, which is the largest anatomic pathology company in the United States, and is really an indicator of both fields moving together quickly in parallel. Not only do you need the test approved, you need to deploy it in laboratories that are seeing millions of patient samples per year, and Quest really indicates how some of the leading labs in the world are now seeing the future is going to be digital, powered by AI, and we're thrilled with those two partnerships signed earlier this year. And lastly, as a sort of complement to our Path Explorer, which focused on the H&E stain, which is sort of the cornerstone of pathology, it's been the cornerstone of pathology for over 100 years, is to augment Path Explorer with a new product we're calling IHC Explorer, which is really the second major class of stains that are ubiquitously available around the world in pathology labs. So this is every time you want to know not only the morphology of a tumor, but also exactly which cells are expressing which proteins. Immunohistochemistry is the dominant platform for doing that. And now pathologists not only can analyze those slides by eye, but they can now analyze it with the power of digital and AI, enabling them to see things like needles in haystacks that you couldn't see manually, or new spatial patterns in IHC expression that aren't available manually. This is important in many areas of oncology, but in particular the area of antibody-drug conjugates, which is one of the most exciting new areas of oncology for helping to cure previous solid tumors that were incurable. So I've already spoken to a lot of this, so I won't go into it in detail, but pathology shortcomings are increasingly addressable through AI. We hear about a lot of areas of medicine where it's very exciting that AI will make, say, incremental advances, but so much of medicine has to do with things that aren't that easy to address today with AI. In drug discovery, as we just heard, there's many steps where AI can help, but AI is not yet advanced enough to actually solve the problem. The beauty of pathology is these are tasks that can be optimized extremely well today for use in pathology, where we can use digital and AI to generate an exhaustive search space of different biological hypotheses from tissue samples. When we have both the tissue sample, the treatment, as well as the outcome, we can advance diagnostic criteria over time in a data-driven way and not just a sort of expert opinion-based way, which has been historically how it's been done. It's super objective and reproducible. One of the limitations of, say, pure human vision is we essentially have a generative model inside of us, and as we're prompted by the same image on different days with different amounts of coffee that day, depending on how angry we are when we're looking at it, frequently different diagnoses will emerge for difficult to diagnose images, say up to a third of the time for difficult diagnoses. The beauty of the type of AI we're deploying for our clinical platform here is it's entirely objective and reproducible and very deterministic, where the same image prompt will always produce the same output. It can systematically learn, not just from image data, but when available from other clinical data as well as treatment data, and can learn longitudinally over time. And it's super scalable. So you could imagine a world where we spend 30 years training the best pathologist ever on the most data, the person who's the best at learning and the best at their field, but that's one person who maybe can process 100 slides a day. The beauty is, using AI and digital, we can learn from massive amounts of archival data. As the system's widely deployed, we continue learning more and more, and then once the models are trained, they can then be deployed at very low marginal cost to laboratories around the world. So this scalability is an incredibly important piece of this that allows us to believe that in the future, the best diagnostics will be available not only to the most resource-rich places, but also broadly around the world to really democratize expertise. So our focus areas of PathAI are really anything that generates a whole slide image, so any area where histology is important, and that sort of boils down to, in the area of drug development, areas like oncology, IBD, ulcerative colitis, as well as liver diseases broadly, but in particular, non-alcoholic fatty liver disease, or MASH. And just to give you one quick image, since this is such a visual field of the problem we're solving in pathology, on the left there is a tiny portion of an H&E stained whole slide image. You can imagine this being something like 1 or 2% of a whole image, but even in this tiny field of view, you can see there's hundreds of different cells from maybe 5 or 6 different cell types. So using AI behind the scenes, we can analyze the entire image, which might contain 1 million cells, classify what every single cell is, what the tissue regions are, and then summarize all that information behind the scenes. So when a pathologist sits down at their desk, they have access to the underlying image, they have access to the single cell resolution heat maps, as well as they have access to the sort of summary statement the AI system has made about the case, which could be diagnosis, it could be a quantitative measurement, it could be a quality measure. Depending on the context of use of our system, we can apply these summary statements to really fit the needs of our different users. So to quickly summarize where PathAI is today, the company really started in translational research, which is the analysis of pathology slides from completed trials or from real-world data sets where we have access to both the images and then in many cases clinical data on the patients or treatment information in drug development. And then we can apply machine learning to learn from the images the characteristics of the patients that are either the best or the worst responders to therapies. Over the past 3 years, we've been deploying our platform prospectively in clinical trials through our clinical development services platform. We're now in over 24 clinical trials in all phases of drug development in oncology, liver disease, and GI disease. I mentioned the Roche partnership in companion diagnostics, which is really the next stage of development. This is really exciting where a new diagnostic test has kind of 1 foot firmly in late-stage drug development to try to lead to the approval of a novel new therapy. But the other foot is in post-approval of a companion diagnostic to be able to distribute that diagnostic globally. And one way to distribute diagnostics is through our digital diagnostics platform called AI Site, where currently we've signed relationships with partnerships, including Quest as one, to process over 5 million slides annually. And we expect this to grow to over 25 million in the next 2 years. So this is my last summary slide, but I just really want to emphasize the really exciting times we're living in with this convergence of cloud, increasingly widespread availability of digital pathology scanners, and then of course the power of AI today, but even thinking about where it's going over the next 5 years, creates this new ecosystem where digital pathology and AI can really be at the core, connecting these 2 regions that have historically been fairly siloed, biopharma, as well as digital diagnostics on the diagnostic side, so we can really picture using digital and AI in translational research to learn new insights, to validate those insights in clinical trials, and then to deploy those insights broadly and globally through the digital diagnostics platform, and then to continue to learn new information from patients around the globe using digital pathology and AI that can then feed new studies in translational research, new trials, with really this vision of increasing the probability of technical and regulatory success in drug development, and then the rapid distribution and adoption of new therapies through digital diagnostics. So thank you for your attention, and I'm going to hand it over to our host.
Thank you.
How are you? My name's Chris Sakalosky. Good to see you guys. I couldn't agree with Dr. Beck more. This is arguably one of the most exciting times we're at. In our point of view, from a technology meets science meets application of those arts and sciences together. When the team asked me to spend a little time and introduce myself and talk about what Google Cloud does, I thought I would do 2 things. Number 1, give you a break from slides. And number 2, not talk about product. We'll have team members around to talk to you about product, but through introduction of myself, I'll actually talk about why I joined the company. You met Dr. Hood earlier today, and so I'll tell you a story. When I first met Dr. Hood, and this is about 5 years ago, this was just after I was asked to found and start up the healthcare and life sciences practice within Google Cloud. And Dr. Hood said, Chris, what are you trying to accomplish inside of Google Cloud? What are you trying to do? And I said, well, Dr. Hood, you have to almost understand the advent of how I got here. Why I joined the company is I got a phone call from one of the presidents inside of Google. They said, we want you to come in and unlock the value of the technology and much of the research that we have done. I said, well, how much research are we talking about? And they handed me an article that was published in The Economist. It was a Nature article, and it showed the top 20 customers, partners in the marketplace that were investing in R&D in healthcare and life sciences. And there were 19 pharma companies on the list, and there was 1 technology company. And the 1 technology company was Google, and it was at number 5. And I said, okay, well, so how much is that? Okay, that's an elite list of folks. We're really humbled to be in it. How much are we actually spending? And it was well over $4 billion a year in R&D. I said, okay, so let me go back to the advent. What are we here for, Dr. Hood? What are we trying to accomplish? our goal was really to unlock the value that was coming from these research pipelines, the work that we were doing in healthcare and life sciences, and bring it into the commercial market so that customers and partners that represent this audience could take advantage of and really fuel. It didn't mean we wanted to go in and create novel drugs. It did not mean we wanted to go in and create digital assets, physical assets for detection. What we wanted to do was unlock the capabilities that we knew would come, as Dr. Beck pointed out, with the scale of the cloud. We knew we were investing heavily there. We could see around the corner in terms of the investments that were coming in GPUs and TPUs, i.e. the advent of artificial intelligence and this generation that we're dealing with today where AI can actually reason. It's not your grandfather's chatbots. It's really a function of, can AI become an assistive technology in the operating room, in the care of patients, in the discovery of new novel therapies, in the detection and personalization of care? And I got really, really excited about that. I said yes, not knowing exactly what I was getting into, but I have taken on that role with such great pride and such humility. I've become one of the largest investors inside the company in organizations like yours. An investment has a wide range of things, but when Bill and I first met, and Bill is actually one of my co-investors in this mission, what I came to realize quickly was that there's a huge community out here. And just to give you a sense for how quickly it's come together since we started investing, I think when you did this first event, Bill, I think there was about 20 people in a broom closet over by our Cambridge offices. And it was just, he put a flyer out there to see how much interest there was in the community. Boston happens to be a great place. First of all, it's us coming back to our roots where a lot of you all are located. But more importantly, there was a small community here. We didn't realize what was behind door number 2, so to speak. And every year that we've had this, it's doubled in size. And this year, it's actually almost quadruple in size based on the community. So we're really excited about that. Our panelists are just great examples of partners of ours that we are investing in real time to make sure that they can reach the broadest markets possible to have the most impact possible. Because Google doesn't like to do things small. Again, we say this humbly, but there's 6 services in the world that touch over 2 billion people on a monthly basis. Google's responsible for all 6. And there's 10 in the world, or 14 in the world that are responsible for 1 billion daily users, or monthly users. And Google's responsible for 10 of those 14. So when we think about touching lives and helping people live healthier, better lives, we think in the terms of billions. And that's not going to be something Google can do by itself. And so we quickly pivoted our strategy to ensure that we were embracing this community that's represented today. So we thank you guys for being here. When we think about the investments we make, cloud's the most obvious one, but people don't realize behind the cloud, the investments that we're making, myself through Bill's team, myself through our broader payer, provider, and life sciences teams, the systems we're building was, let's pull friction out. Where can we pull friction out of the ecosystem with cloud? Building data platforms so that you can build longitudinal views of a patient. That was a big first step. That was actually one of the first things I asked the company to build for our sector. Let's build this longitudinal system, this data platform that you could run longitudinal large diagnostics off of the top of. That was something big. Right behind it, you heard some of our speakers talk about the modalities. Okay, well, data doesn't come in one format. It's not just text. It's images. It's a broad form. How would we then accommodate that? So we started building the platform technologies for imaging. We started building it for diagnostics. And so we have an entire suite of technologies purpose-built from data platform on up for the healthcare space. We're the only hyperscaler that has put that much end-to-end work into the platform, because it's not about the cloud, it's about what you can do with the data on top of the cloud. You bring that hyperscale to the work that we're doing. And so we're very, very excited about that. And then we started putting it in the hands of our customers. Where could we take large language models? Where could we actually build some of our own first party? And you're seeing those coming out now. One of the first of them was AlphaFold. We're pretty excited about that one. We got a couple of Nobel Peace Prizes in the door, and we're super proud of the researchers that were there. We've made that available via open source, and you'll constantly see this sway inside of the company, putting things out in open source to get broad scale. I think AlphaFold, Bill, I think it's 50 million users, active daily users using AlphaFold across all the cloud platforms. We do that because that's our broad contribution to the scientific community. But when we are also putting out very purpose-driven models, things like MedLM, things like imaging LM for cancer and lung screening. Why are we putting those out as first party? No, we do not expect to heavily monetize those. It's not our intent. And there's a great debate inside the company whether specialized models versus traditional foundation models will eventually become smart over time. My vote's in the middle. Yes, I'm an independent, so I can't tell you who I'm voting for in the presidential election. But I kind of come up the middle of them, the two. I think you're going to have both. I think you're going to have specialized models that are brought by Google and our partner ecosystem that those large foundation models call as experts. I think that's been coined the term MOE, multitude of experts. I think that's where you'll ultimately see the market settle in. But we're very, very excited to invest not only in our first-party models, but as importantly in our third-party models. And we've built a model garden that is discoverable to the entire world. And I think we have the most health-based models in our system today. And we're launching several more that you'll see in the coming weeks. And so we're very excited about those and bringing them to the market with you. And it's with that context when we talk about the power of AI, the power of large foundation models, and the power of something I see coming around the corner with agents and agentic search. If you're like me, I had to look up what agentic meant when I first heard it because I didn't actually correlate the fact that it's the next generation of artificial intelligence wrapped around a chatbot that can allow you to reason. It puts a co-pilot in your pocket, whether it's a doctor in your pocket, it's a researcher in your pocket, whether it's a notebook that allowed you to ingest tons of information, hundreds of years of information, and actually create a podcast that actually you can interact with in natural language to ask questions about what's the most likely next secondary use for a particular chemical compound or therapy. Watching our customers take these technologies that we built and putting them into use in practice, as you saw, is pretty exciting. So without further ado, I'm actually going to call our panelists back. We're going to do a fireside chat with you. Again, I'm going to give your brains a break from slides. I'm going to call our panelists back to the stage. And we're just going to do a little bit of Q&A on what they're seeing and how AI is impacting their businesses. And quite frankly, the mission that we're all here, which is trying to predict and ultimately eliminate cancer from this planet. So thank you. Donna? Doctors, thank you for joining us again. Appreciate having you guys. It's really fun to hear your conversations. Listen, I'm going to start with a quick jump ball question, and we can kind of go in any order you'd all like. We're 3 to 5 years out from now, and we're reading a headline on artificial intelligence. It's either something that's impacted your company or it's some profound impact that artificial intelligence has had on this sector, and specifically in oncology and cancer. What's the headline read? Dr. Beck,
sure.
I'll jump to you.
I think an exciting headline would be, or fact at the end of the year, something like more than half of new drug approvals have an AI-powered companion diagnostic, and more than 90% of diagnostics around the world are leveraging digital pathology and AI. I think that is achievable and would be amazing.
Dr. Lin?
I'll be aspirational. We'd hope to launch a multi-cancer screening test by then. So I would love the headline to be, thousands or tens of thousands of lives saved through annual testing through blood screening.
Working in small molecule drug discovery, I'd like all the headlines to be about the success of the therapeutics and the benefits patients are seeing from them. We're not developing these technologies for the sake of developing the technologies. We're developing them to intend to help patients. And having better drug therapies for unmet need, that I think is what I would hope the headlines are about.
All right, well, I'm gonna look at the papers, the digital papers in 3 to 5 years and see if any of these things have come to life. All right, so I'm gonna kind of go in order. Dr. Abel, I'm gonna start with you. Given our talks around artificial intelligence, what are your thoughts on how AI can overcome some of the limitations to traditional drug discovery today and the methods that we see and develop more effective and targeted cancer therapies?
So the majority of drug discovery today is done through empirical methods, and those take way too long, they're way too costly, and they fail to deliver a good drug therapy much more often than we would all prefer. The hope is predictive modeling, machine learning, artificial intelligence. These things can work together to improve our success rates and allow us to more rapidly and more efficiently deliver drug therapies to patients that will succeed. So that's really the core value proposition of these technologies as we see it.
Fantastic. Dr. Lin, over to you. how can we ensure AI and the algorithms that we're using, that are specifically used in cancer screens, are trained on diverse data sets? And because we've been talking about data throughout the course of the day, how do we ensure that they're trained on diverse data sets and then accurately reflect the broader population? What's your take on that?
Yeah, I think what we've learned really, I mean, everybody here, I think, AI, really the advancement that really sort of propelled even the image side, right, is ImageNet, right? It's not necessarily the algorithm, yes, back-propagation and also, attention-based systems, but the data ultimately is what's, really sort of driving it on top of the compute there. So getting the right data sets is really, really critical. So for a lot of our studies here that we did at Freenome is, again, making sure that the representation of data is not only, people who sign up, but working with building trust with minority parties and groups and universities there to not just sort of jump in and says, here, give us your blood and give us your data, but to establish trust so that we can have data sets that's representative of not only sort of US population, but also global population there. But we need to do it very carefully, not only establish trust with the communities, but also ensure all the things with privacy and protection that's going to be really, really critical. So I think as the whole field starts to generate more and more data, I think this is a very, very critical aspect we should think about.
There's a lot of learnings in there. One, pick your partners wisely, because the data set that they have, I mean, look at it and get a good sense for who their partners are, where the data was acquired. That's a big one. And then obviously in their own trials, in the work that you're doing out there, making sure that the data is diverse, obviously will open up our corridors. Thank you. Dr. Beck, digital pathology is an emerging sector for us right now. I think we're probably at the early stages of seeing the impact. And you guys have been a leading voice in this. How's this new era of large language models? How is that expected to impact the area, and how are you thinking about this at PathAI?
Yeah, no, it's a great question. I'd say we think about it in two big ways. On the one hand, when we founded the company, and still today, a lot of the issues were solving the challenges with manual scoring. I think I said it in an offhanded way, a lot of what, the human brain is generative in an amazing way, but different times a day, the same prompt will result in a different answer, and if you have different people with different models in their head, the same prompt will often also result. Darth Vader over here will also result in a different answer. So a lot of the core problems out of the box with LLMs is they're not always super reproducible. They're very good at seeming confident when they're wrong. They're superficially seem very definitive when underneath there's not, in all cases, a ton of evidence for that seeming confidence. So they have a lot of the same problems as humans, for better or worse. And there are certain tasks that they're extremely good at, which are ones where it's very important to be human-like. But sometimes it's just better to be machine-like. So our task in pathology is to actually make anatomic pathology more like clinical pathology. So if you submit a blood test somewhere, you don't have a ton of fear that you're going to get a wildly different answer if you submit it to a LabCorp or a Quest in Boston versus San Francisco. Like, that's been very highly reproducible, highly deterministic. You might say it's boring, but you know what you're going to get based on what you put in. Anatomic pathology is just not like that. The irreproducibility rates are far too high. It's not objective. So to some degree, the core challenge we have is how do we make these models so good that a human plus the AI system, we drive error rates down to 0, we drive reproducibility rates up to 100%, and we provide it with the background knowledge, which LLMs can certainly help with for summarizing the literature of, based on this diagnosis, this is a treatment recommendation. And that last piece is important. So I think it's an important adjunct. Our core value prop is objective, reproducible, deterministic. LLMs don't seem to play a huge role there. But then how do you improve the ability to connect this answer with the rest of the medical literature, which we all know is growing every day? LLMs are transformative there. And then how do you not only collect the literature, but then point physicians and patients and people around the world to what action should be taken next? And how do we help oncologists with that? LLMs are very important there. So it's important, I'd say, downstream, and some of that will build into our product, and some of that we'll work with partners on.
Great, thank you. Dr. Lin, I'm going to come back to you. What are the most promising AI applications that you are seeing for detecting precancerous lesions or early stage cancers that are often missed by traditional screening methods? What are you seeing emerge in this space?
I think there are some methods and modalities that already exist, right? So since Illumina is a partner, right, looking at nucleotides is something that's very promising. Proteins are very promising, but very hard because proteins is unstable and they're, in terms of order of magnitude, very different. There's looking at also autoantibodies, and there's look at different proportions, right? Extracellular vesicles. I think we're just starting really to understand what is actually available in the blood. We're still only looking really at the data underneath the lamppost. And we need a lot more lampposts there. And I think Illumina has done a great job of sort of establishing a lot of data, but I think there's going to be a lot more technologies that's going to be needed. And I think the key is to how do you think about how these different technologies overlap? And that's why we're sort of multi-omic. We're agnostic to one single technology. And some cancers will be really good using a protein technology. Some will be really good with a methylation technology. Some will be very good for nucleotide-based technology. And how do you are able to really sort of combine this in a test? And that's sort of the thing that we're really looking at all the newest technologies that are coming out. And don't think that every cancer is the same. And be able to then sort of generate the data, to be able to generate the evidence, to be able to really sort of figure out which one is the best for which cancers.
Any inside baseball, anyone, obviously beyond Freenome and Illumina, but anyone, any new technologies emerging or any new players out there that you're impressed with that you're talking to today?
Probably the ones we're talking to probably wouldn't be able to share. But I think the key here is, I think, the traditional methods, for example, like mass spectrometry, or immunoassays on the protein side, or just generating more data for those, I don't think it's going to be sufficient. So we need new modalities and new dimensions of looking at what's in the blood.
Sounds great.
So those what's going to be sort of exciting, whether proteoforms, right? Looking at how proteins are different, looking at different types of chemical modifications for DNA. So I think there's whole new dimensions that haven't yet been explored.
Excellent, thank you. Back to Dr. Abel. What are the biggest challenges in integrating AI into your existing drug discovery workflows? You have significant investments, as do most of our customers and partners here in the audience. What are some of the challenges and how are you addressing them?
Yeah, so for Schrödinger, it's easier than most other companies because Schrödinger was founded on the idea of predictive modeling being able to drive value. So our whole setup was built from the ground up to allow predictive modeling to guide what we do. But challenges do come up. So oftentimes you have a really interesting idea you want to explore, but it requires burst utilization of a huge amount of compute, and you have to forecast what the return on investment of that is going to actually be. So there's all sorts of like practical issues that come up in terms of using it on a day-to-day or project, over the arc of a project kind of way. But we don't have sort of the organizational cultural barriers that typically come up internally. Where that does sometimes come up is in our interactions with collaborators, where to allow these methods to really create as much value as possible, they need to be put in the position to really drive the decision making on the project. And that's a really big shift for a lot of organizations when they start collaborating with us. So the way we try to have that conversation is in a very data-driven way, that there are periods in collaborative projects where we'll just keep track with enterprise informatics systems that actually are cloud-driven as well, just how well the predictions are for the predictive modeling versus the traditional human-driven strategies. And then if you can show people a dashboard over several weeks or several months where you can compare these things head to head and allow people to see themselves where the incorporation of greater predictive modeling can allow for greater acceleration of their discovery projects, we often find then the project teams in a fully collaborative fashion will then move over to allow these methods to be used more in the driver's seat. But it has to be something where you're showing people how well they're working rather than telling someone you should be doing it differently. It's much more of the data-driven strategy for us at least has been much more successful.
Got it. I'm going to come over to Dr. Beck. So a colleague of mine, Bakul Patel, he came over from the FDA. He's been working with Google now for about 4 years. He and I often have conversations about this emergence of AI and how it's going to play, whether it's in software as a medical device or whether it wakes into some of the new methodologies and methods that people are doing, everything from early detection to discovery to the broad therapies themselves. Question in that realm, What are the implications of the regulatory approval for AI products in the digital pathology space? How are you embracing the work, because we want this to be a safe ecosystem? At the same time, there's no true rules wrapped around this. So how are you embracing that in your space?
I'd say maybe 3 different ways. One, we very much, and I think one thing that's been pretty unique about our company is we see AI almost like electricity. Like the most important thing is what problem are you solving for the user, and the infrastructure, the integration, the workflow, and then how can we use AI to make the current process better? And some of those processes at the end of the day are highly regulated by the FDA and others aren't. And we want to really work with the FDA and regulators on both, but use AI for everything from identifying artifacts, helping to prioritize cases, helping pathologists view the portion of the image they should view correctly, helping to report with deep integrations into things like laboratory information system and EHR. And then the FDA has been thinking about how to regulate medical devices for a very long time, and I'm very much a proponent that the way to do this that's most efficient and certainly safest and most effective for patients in getting devices that are important for them approved is to really think about the product at the end of the day. What's the input and what's the output? And that often the FDA is very comfortable with regulating different diagnostic devices, workflow devices. So we always try to put it into that framework and to work closely with the FDA as very close partners on some of our most diagnostic tools, but other things aren't really in the FDA's purview that are much more on the efficiency pure workflow side of the spectrum. And the other part of my answer that I think is very, very important in our case is we are aiming to be an adjunct to the pathologist and, on the clinical side, and not to replace the pathologist. So it lowers the bar tremendously in terms of us getting things out quickly, because we just have to show that this is the performance without using AI, and this is the performance with the pathologist using AI. And we think that allows us to really build our tools to optimize the things where the combined system is going to be clearly superior to the system not leveraging AI, versus trying to both recreate everything the pathologist is doing and then do it safely and more effectively, which is just a much, much higher bar to cross when your real goal is ensuring safety for patients.
Yeah, we've seen a great embracing of technology, not only from the panelists and our partners in the audience, but even with the bigger tech companies like Google, from Dr. Califf over at the FDA. And I think that will continue to accelerate. I think he understands technology in his core, but again, we'll take our cues from him, but continue to push the thresholds and bounds. All right, I know we're coming right up on time, so I want to make sure I keep us on. So I'm going to ask sort of a follow-on jump ball question for each of you. If you had one bit of advice or one call to action for the audience, what would that be? And we'll just come right back down. So Dr. Beck, back to you, and we'll come right down.
Just the criticality of diagnostics for ensuring effective therapies get to patients, and then ensuring post-approval that patients globally have access to those medicines. We spend so much as a healthcare system on the therapeutic side, but equal attention, or maybe not equal dollars, but certainly equal attention, if placed on the diagnostic side, can have a huge impact on the probability of success of development and then the successful commercialization of therapies for patients.
Fantastic. Dr. Abel?
Call to action. It's interesting, the pathology comment. I would say these technologies are going to teach us more about biology, fundamentally. And as we learn more about the biology, that also opens up new avenues for therapeutics discovery. So rather than sort of a division between the two, I hope the two can work together over time to allow us to better understand the biology, so we can have better drug therapies, and also through better detection systems, figure out the patient populations and the people that need those therapeutics more readily, so we have better prescribing of those medications. So I think over time these will all be self-reinforcing in a way that will be really helpful to patients.
Thanks. Dr. Lin?
Obviously, AI is a huge field, and AI is a lot of things to a lot of people. And I think what's probably most important in terms of solving the problem is actually defining the problem up front. What is actually the clinical difference that you will be making, and why is that important? How is that going to sort of change how healthcare is either delivered or being used? And then talk to the sort of right stakeholders, then start figuring out what are the tools that's going to be important, what are the data sets that's going to be needed to be generated, and how are you be able to sort of get those pieces together. So I think ultimately, defining the problem and understanding the problem is probably going to be the most important, because there are so many uses of AI it can be, and really have a problem-centric approach is going to be very, very important.
Great. Listen, I'd like to thank our panelists not only for their enlightening speeches to start, but for the insights and calls to action to wrap. So thank you very much for the time. Appreciate it.
We've tried everything. There's nothing more that we can do. I heard those words, but between my organs shutting down and the unbearable pain, I didn't really know what my doctors meant. And then my family came into the room to say their final goodbyes, and a priest read me my last rites. I was 25 years old. Just 6 months earlier, I was a former college quarterback and a medical student training to become an oncologist in memory of my mom, who had died from cancer just a few years before. This week actually marks 20 years since her passing from brain cancer. I had promised her that I would dedicate my life to trying to find treatments in her memory for other patients like her suffering from cancer. And then there I was, literally dying in the ICU from a completely different condition. At the last possible moment, the diagnosis was made. Idiopathic multicentric Castleman disease, a rare disease where your immune system attacks and shuts down your vital organs for an unknown cause. 1/3 of us will die within just a few years of diagnosis, and the other third within 5 years after that. Unfortunately, there were no FDA-approved drugs, but in a last-ditch effort to save my life, my doctors gave me a combination of 7 chemotherapies. Somehow I survived, but I would go on to relapse and nearly die multiple times thereafter. I was able to return to medical school at Penn and finish my 3rd year of med school while on an experimental drug, a drug called siltuximab, that we hope would keep my disease in remission. It blocks interleukin-6, and we hope that this could maybe keep me well enough to finish medical school and continue my dream of treating patients in memory of my mom. But unfortunately, I relapsed, and I nearly died for a 4th time. What was so difficult about this relapse is that I was on an experimental drug, the drug that we hoped so badly would keep my disease in remission, and it didn't work. My doctor came in the room and explained to me that there were no more drugs in development, there were no more promising leads. Just like before, when he told me that we'd run out of options and he gave me chemotherapy, we'd try chemo again, but he told me that at some point the chemotherapy is gonna stop working and I'm gonna die from this disease. And I just couldn't believe that we didn't have any promising leads, anything out there that could maybe treat my disease. So my hope really became, could there be a drug that was already FDA approved for another disease? Maybe it hadn't been tried for Castleman, but that could maybe be repurposed for me. This concept of repurposing is something that all of you in the oncology space know very well. Drugs are tried for many different cancers and many different non-cancers all the time, but unfortunately it's not done systematically, and there's some really great examples of drug repurposing. And of course, in my case, the reason I needed to focus on repurposing is that I didn't have $1 billion in 10 to 15 years to develop a new drug from scratch. If I wanted to survive, I would need to find an existing drug that could be repurposed for me. And as I said earlier, there are so many great examples of repurposing. A drug like Viagra, which we all know was repurposed from heart disease to its well-known use, but most of us don't realize that it's also been repurposed for a rare pediatric lung disease. Kids that were dying as children are now living full lives on Viagra. In addition to that, there's thalidomide, which has a horrible history of causing birth defects, people being born without limbs. Now patients with leprosy and also with myeloma can live for years after their diagnosis because of thalidomide. One of my favorite examples is tocilizumab, which many of you all will be familiar with in the cancer space. Toci was actually made for Castleman disease by my dear friend Kazuo Yoshizaki. Kazu discovered that interleukin-6 was important to Castleman disease, and he developed a drug to block the receptor for IL-6, tocilizumab. And when I was talking to a colleague of ours, Makoto, about this, he said, you know Kazu gave the drug to himself as the first patient ever. I said, Kazu, is this true? Makoto just told me you gave yourself tocilizumab as the first patient ever. He said, no, I didn't give it to myself. The nurse gave it to me. Oh my gosh, he discovered the target, developed the drug, and then he gave it to himself. And when he didn't die, he went on to develop it for Castleman's disease, where it got approval. But many of you all will know tocilizumab in the cancer world, because about 10 years ago, when colleagues of mine at the University of Pennsylvania were treating the very first pediatric patient with cellular immunotherapy, she experienced a cytokine storm that looked very much like Castleman disease. And my colleague Carl June gave her tocilizumab, this drug that was made for Castleman disease 20 years before, and it saved Emily Whitehead's life. In doing so, it saved the entire cancer immunotherapy program, because if the first pediatric patient to ever get cellular immunotherapy would have died from a cytokine storm, the whole program would have been shut down. So another example where a drug that was made for one use, in this case Castleman's, could be helpful for cellular immunotherapy. So here I am with this deadly disease, Castleman's. I'm told there are no more treatments out there, but I'm just thinking to myself, could there maybe be a drug out there that was made for another condition that could actually work for me? And every time I walked past my pharmacy, I would look at the pharmacy shelf and I think to myself, is my treatment sitting there on the counter? Is it right there? Is there a drug there that was made for something else that could maybe be life-saving for me? I was able to finish up medical school. This is me on my graduation day with my fiance at the time, Caitlin, and I dreamed about the idea that I could go on to continue to advance treatments and hopefully treat patients in memory of my mom. I enrolled in business school shortly thereafter, recognizing that many of the challenges for progress had nothing to do with science or medicine. They were organizational challenges. So I made it a couple months into business school before I relapsed and then nearly died for the 5th time. And this relapse was really tough because I was getting weekly chemotherapy leading up to that relapse. In fact, between my 4th and my 5th flare, I actually was diagnosed with a rare form of liver cancer and had that cut out. So I was really, really hopeful that maybe with the cancer resected and on weekly chemotherapy, that Castleman's would not come back. It still came back. I spent a month in the ICU with this 5th flare, somehow just barely survived. But when I left the hospital, it was different this time. And that's because I'd been collecting blood samples of myself every week leading up to my relapse. Because I thought if I did relapse, I'd want to have some samples and maybe I could do some experiments on those samples. And so I had a really simple concept, and that was if we could understand what mechanisms were going wrong in my immune system, maybe there's a drug out there that could maybe treat it. And so I performed first a series of cytokine panels where I discovered that vascular endothelial growth factor, or VEGF, and SOLVAL2 receptor, which is a marker of T cell activation, were highly elevated in the weeks leading up to my relapse, which was really interesting, and maybe we could target VEGF with bevacizumab or maybe deplete my T cells. But importantly, and then also we confirmed there's tremendous T cell activation, but importantly we wondered, could there be some sort of final common pathway that's involved in the T cell activation, the VEGF, that can maybe be targeted? And so I ran serum proteomics, measured over 1,000 serum analytes, and detected a really strong mTOR signature. So mTOR is a critical communication line in the immune system and beyond that could really fit the bill here. So I thought, well, maybe mTOR is that central common pathway, and if we could target mTOR, that could save my life. And did an experiment where I tested mTOR activation in a number of individuals that did not have Castleman's, a bunch of normal lymph nodes. And in this particular image, if mTOR is turned on, then the cells will stain brown. If mTOR is off, they'll stain blue. And you can see that in a normal lymph node that there are certainly some cells that have mTOR turned on. And then I stained my lymph node, and I was just blown away by the tremendous amount of mTOR activation in my lymph node. So hypothesized from the proteomics, but now confirmed in my tissue. So I thought maybe we could use an mTOR inhibitor like sirolimus, which has been around for decades, and maybe that could keep me in remission. And I've just crossed about 10.5 years that I've been in remission on this drug. Oh, thank you, thank you, thank you, thank you. So now it's been 10.5 years in remission. I was able to marry the love of my life, Caitlin, have these 2 beautiful kids, and it just feels like such a dream. Never could have imagined that I could be here. During this time, we were able to raise a lot of awareness about this concept of repurposing. The New York Times called it "Doctor Cure Thyself," which I think is a bit of an overstatement. I think it should be, I hope that this lasts for a long time, but I don't know if I want to use the word cure. I also wrote a book about my journey called Chasing My Cure to get the word out about the fact that this drug was just sitting there waiting to be utilized. About 10 years ago, or about 9.5 years ago, I joined the faculty at the University of Pennsylvania to start a center called the Center for Cytokine Storm Treatment and Laboratory, or the CASTLE as we like to refer to ourselves. In the last 9.5 years, we've unlocked and advanced 13 additional repurposed treatments for diseases they were not intended for. So these are 8 patients of the thousands that are alive today because of drugs we found they could be useful in ways that they were not made for. I just want to highlight a few patients that really stand out. So one of them is Michael. Michael came to us in 2016 with a rare and horrible form of cancer, angiosarcoma. And he asked, well, first off, he had failed to respond to the 2 drugs that were used, and he asked, is there something else out there that can maybe be useful for me? And we did the simplest thing ever, but this is the right place to bring this up. We Googled angiosarcoma treatment. And we found this is the first time I've ever given a talk where I can say that, but so yeah, go Google, go Google. It was a very sophisticated algorithm called Google. And so we Googled it, and what we found was that there was a paper published 3 years earlier indicating increased PD-L1 expression in a handful of patients with angiosarcoma. Now, no one had ever given anyone a PD-1 inhibitor at this stage with angiosarcoma, but based on the fact that there was increased PD-L1 expression in these few patients, we thought maybe PD-1 inhibition could be useful. Now, this is 2016, before everyone got PD-1 inhibition. And so we confirmed that this patient's tumor had increased PD-L1 expression, and we treated Michael as the first patient ever with a PD-1 inhibitor with pembrolizumab. And Michael has been in remission for over 8.5 years on pembrolizumab. Just so incredible. It's just amazing. And actually, just last month, Michael's son got married, and Michael was able to walk his son down the aisle, which is just amazing. Equally as importantly, based on how well this drug worked for Michael, and beforehand everyone uniformly died from metastatic angiosarcoma, other patients started getting it, other doctors started giving it out more frequently, and without ever doing a randomized controlled trial, it is recommended by the National Comprehensive Cancer Network as the first-line treatment for metastatic angiosarcoma, again, without requiring any clinical trials, because it was so deadly before, showing that it could work changed the entire paradigm. I also want to highlight Allie's story as well. This is not in the cancer world, but Allie has a rare condition called DATA2 syndrome, where kids have strokes until they die in their teenage years from having literally hundreds of strokes. And importantly, about 20 years ago, a doctor tried a TNF inhibitor in a patient with DATA2 syndrome, and that patient stopped having strokes. But it took about 20 years between using this drug for the first time and us collaborating with a group to establish treatment guidelines on how do you treat DATA2. So now we've come out with guidelines so every kid diagnosed with DATA2 gets a TNF inhibitor. But 20 years went by where the world knew that a TNF inhibitor could stop strokes and save these kids, but people weren't getting them. So this just blows my mind. The answer was out there, but we weren't acting on it. So just want to frame this briefly. There are about 4,000 drugs that are approved for about 4,000 human diseases. But we have an additional about 14,000 diseases that don't have a single approved therapy. And now the way we usually think about things is let's develop new drugs to address both the diseases that have drugs and the diseases without any. But actually there's incredible potential to use the FDA-approved drugs we have for the diseases without any. So every time I walk past the pharmacy, I just wonder how many more life-saving cures are sitting there that could help patients right now that are suffering from horrible conditions. The question is, why aren't they utilized? Well, first of all, it's financial. There are no financial incentives for the vast majority of FDA-approved drugs that are already generic to find a new use. You can treat another 1,000 people, but if it costs $1 a pill, you can't justify doing the additional work. Additionally, there's never been anyone responsible for making sure drugs are fully utilized. So a couple years ago, I launched a nonprofit called Every Cure, along with my co-founder, Grant Mitchell and Tracy Sikora, that's on a mission to unlock the full potential of each FDA-approved drug to treat each and every disease and every patient that can possibly benefit from it. So in doing so, we are doing what's often called drug repurposing, but we think about it a bit differently. So traditional drug repurposing, you start with a disease like Castleman's, and you look across all the drugs, and you say, is there a drug out there for Castleman disease? Or traditional indication expansion, if you're a drug company, you start with a drug, it's approved for one disease, and then you say, are there any other diseases out there that my drug could be useful for? What we're doing, we call computational pharmacophenomics, which is we start with all diseases and all drugs. We quantify the strength of evidence between every single drug and every single disease, and that enables us to identify the most promising opportunity to repurpose a drug for a given disease. It's not necessarily the disease of interest or the drug of interest to you or to your company, but it's the greatest opportunity to help people, period. We do this by training or by utilizing biomedical knowledge graphs and training them with a handful of machine learning algorithms, my favorite of which was developed at Penn State. And so we train on known treats relationships, and we apply those algorithms to the rest of our biomedical knowledge graphs to come up with a single score from 0 to 1 for the likelihood of that drug to treat that particular disease. And so the underlying framework, as I mentioned, is a biomedical knowledge graph. We have a handful that we utilize. We also are thrilled to accept data from any life science companies or data science companies in the room today that can be contributed towards our biomedical knowledge graph and the work we're doing as a nonprofit. We generate a treat score for every drug versus every disease. It's about 75 million scores. Within a year, we're going to be sharing these scores publicly with the world. So you can look at your favorite disease or your favorite drug and see every ranking for that. We use our listing to identify the promising opportunities that we want to do further work in, that we want to validate in the lab, we want to do RWE studies in, and eventually that we want to take into clinical trials, with the goal of ensuring that patients can access the drug. And in some cases, it'll result in FDA label change, but not in all cases, because we're only talking about FDA-approved drugs. Between 20 and 30% of all prescriptions written every day in the US are off-label. So doctors are prescribing things for diseases they're not approved for all the time. We're here to put good data behind it. I want to highlight 3 archetypes briefly. So when we talk about repurposing, there's the group that we call a frontier explorer, which is like sirolimus for Castleman disease. Early insight that it might work, but there's a lot of work to be done. You got to work in the lab, you got to do clinical trials, then you got to increase utilization. We have what we call the clinical gem, which is like pembrolizumab for angiosarcoma, where there's a good mechanism, there's clinical data, but we had to do additional clinical work, in that case real-world evidence, to really establish that the drug works for that disease. And then finally, the unsung hero, like this TNF inhibitor for DATA2, where the mechanisms sound, the clinical data sound, but it's just not being utilized because there's not enough of a financial incentive. We also want to take those forward to make sure that people aren't suffering when the data's out there. In my last minute or two, I want to highlight some results from the first run of the algorithm about a year and a half ago. We looked at across, of course, many of the top hits across many diseases, but we also zoomed in on Castleman disease given our focus. And we were really interested to see adalimumab as the #1 hit for Castleman disease because it had never been used before in Castleman's. But right around that time, we had a patient named Alan in Vancouver who was failing to respond to all therapies, including the one that saved my life. And he was getting ready to be transferred to hospice. In fact, the order had already been placed. And we recommended trying adalimumab for the first time ever in any patient with Castleman disease because we were out of options and we didn't know what else to try. And Alan responded incredibly well to adalimumab. It's now been over a year and a half that he's been in remission. He didn't have to say goodbye to his daughter, and I'm just so excited. It was an early N=1, but this sort of highlights the power and the potential of an algorithm like this. I also want to highlight Joseph's story really briefly. Joseph, in January of this year, so a little bit under a year ago, was battling a rare disease called Poem syndrome. I was getting ready to again be transferred to hospice care when no chemotherapies were working. We looked in our algorithm. Well, first off, we were contacted by his girlfriend, looked in our algorithm and identified 3 drugs that scored very highly for Poem syndrome, drugs that actually were developed for multiple myeloma, and recommended them to his doctor over a weekend, and who tried those drugs, and Joseph responded really, really well and is doing great. These are early examples of specific drug repurposing opportunities for specific diseases, which is, I like to think about it as the lowest hanging fruit on a given tree. You know, what's the best opportunity for Poems or Castleman's? But what I think is really exciting about what we're doing with Every Cure is not just to find the best opportunity on the Poems tree, but what's the lowest hanging fruit across the entire forest? What treatment opportunities are out there that are just waiting to be unlocked if you do it in a disease and drug agnostic way? We've got some amazing partners that have helped us to get this far in the last couple of years. Hope many of you all in this room will want to join us on our mission. Earlier this year, ARPA-H awarded us about $50 million to build out our AI platform further. And then just a couple weeks ago, TED, their Audacious project, just announced additional funding to take all those really promising predictions all the way through patients and reaching patients all over the world. Had a lot of support. Not to get political, this is President Biden's Chief of Staff, Jeff Zients, the White House, and many throughout the federal government, and really excited about the progress that we're making. Got an amazing team. Really excited about adding Janet Woodcock to our board just a couple weeks ago, but we were really, really fortunate. So I just want to close. My last slide is just highlighting future directions. As I shared earlier, we would love to partner with every single organization here that has access to data sets that can inform drugs, diseases, targets, mechanisms, can be fed into our knowledge graph. Would love to think about ways to work with you on improving our algorithms. We've got a handful of great algorithms, but can always get better. And would love to think about ways that we can validate the most promising hits. And what I hope we can work towards is a world where no one is told we've tried everything when there's a life-saving cure sitting on the pharmacy shelf. Thank you guys so much. Yep, here we go.
Great job. That was awful.
Thank you.
All right, so can you hear me? Are we hot? We are good. So I'm Will Morris, as I take out my panel. First of all, no one's talked about the cyclorama that we're in, right? It's the world's smallest carousel, apparently. Pretty impressive space, but do you guys know what the derivation of the word cyclorama is? Like what it's intended to do or mean? So 1884 was one of the world's largest domed next to the Capitol, but a 360-degree picture is the lexicon, is the word origin, which I actually think is unbelievably appropriate for today's summit. It's a 360-degree picture. Ideally, the patient, the provider, the researchers at the center, but it is about all of us kind of encircling and providing different perspectives, unique capabilities. I think that makes this moment unbelievably special, and thus I think it's actually quite appropriate and telling to kind of be in this lovely space. So we have 6 physicians here, no copay. We have 30 minutes, which is less time than it takes to do prior auth. So we're gonna have to be brief. That's it for me, I promise. So we already know David, that was a tremendous talk, but maybe you can talk a little bit about every cures in kind of, especially for the lens of the clinician. So we're gonna be talking about kind of, yes, AI and the practitioners, those that use, but a very diverse set of clinicians here representing different aspects, think the 360 degrees, that it takes to render clinical care.
Sure. So as I mentioned briefly, we're generating a score for the likelihood of every drug to treat every disease using the world's biomedical knowledge. And the intention, the primary intention, is to identify the most promising opportunities to move them forward. But we'll also be sharing all these scores with everyone. So if you're a clinician and you have a patient with Poem syndrome or DATA2 syndrome, or you name the rare condition, you'll be able to type in the name of that condition, and then you'll be able to see a rank ordered list for all 4,000 diseases and the likelihood, all 4,000 drugs, and the likelihood that every drug could be useful for that disease. And I'm so amazed that adalimumab, had never been used in human history for Castleman's, and that was the prediction that was made, and that saved this first patient's life. I don't think it's always going to be the first one on the list. I think that if you're a clinician, I don't recommend you say, oh, number one on the list, I'm just going to give it to my patient. But I think that combining together the world's biomedical knowledge, these sort of scores, and then also your clinical judgment and abilities, I think it's going to help to give you a bit of a multiple choice answer as opposed to a fill in the blank.
I love it. Sanjay, maybe you can, from a practitioner, clinician, oncologist in the community, also quite the large social media. MySpace, is that right? No. And now at Tempus, but maybe your perspective, and tell us a little bit about kind of yourself and your journey.
Well, hearing the talk previously that you'd given, it kind of made me realize one of the issues that's really hard in the sense of like a moral injury aspect of a community oncologist or an oncologist in general. And that's when you are talking with a patient and you're kind of giving this sometimes wide range of likelihood of response. So it's like 40% to 63% chances, chances of working. So either likely to fail or likely to work, and that's my range. And science doesn't know what it doesn't know. We know that there are these kind of point considerations on things that may actually influence that. There's a lot of study on, proteins and even on a DNA level of what may say, okay, this is maybe why not. But there's stuff on metformin and statins and polymorphisms and all of this variability that's very hard to, unless you aggregate it, really know where someone is at that 40% or 65%. And what's worse is when you have something that's favorable and you're high-fiving and there's tears of joy, and it's 85% chance of working, and it turns out it didn't do anything. Yeah. And so this moral injury, you kind of dial up and down on really the known and unknown. You know, studies, if you know your patient is underrepresented on this study, and do you share that? Do you just say, well, we're going by this data even though it's not really reflective of your, you know, cohort? This is where I think the aggregate of experience is very valuable. I mean, any person that's gone through a cancer diagnosis, you know, oftentimes a lot of my friends and family says, hey, who's the best person for this? And the reason you're asking who to go to is really predominantly for two reasons: what they know and their experience. Yep. And they want their experience because they want the best predictive aspects of outcome, whether it's toxicity from treatment or from likelihood of a success. And I don't need to tell anyone here that that's very much what, you know, AI and machine learning is hoping to do, is prediction on a set of variables at a depth that is, you know, quite challenging for a human brain anecdotally to appreciate or even cipher through, you know, on a very organized study.
So I'm going to go to Bob next, because I think there's a natural kind of progression of the story, so bear with me. You know, you've got the clinician who's kind of grasping at all these, and Bob, maybe you can explain a little bit from US Oncology and Ontada at McKesson, but moreover, kind of, you're a pane of glass, you're an experience layer for the community oncologists, and, you know, electronic medical record, a place that you is a repository of data, or is it something that can be more of a utility that actually assists, not guides and doesn't replace the clinician, but assists the clinician in the practice of medicine? So
yes, so that is the interface where I lived. I was a practicing hematologist oncologist for 26 years in Northern Virginia and US Oncology. Ontada is tasked with harnessing that data for two purposes: continue to improve the data and support technology to assist physicians in practice, and at the same time take the data that is generated in the usual care of cancer patients to make that available to pharmaceutical companies, mostly in the vein of real-world evidence studies supporting post-approval data requirements of pharmaceutical companies. So one example where we have provide decision support to the day-to-day practitioner, physician, nurse practitioner, et cetera, is in the ordering of chemotherapy. The physician has to follow guidelines that are put forth by the National Comprehensive Cancer Network. These are both in accordance with the drug's label, as regulated by FDA, but also some off-label use that's supported by evidence. As the physician enters the diagnosis and prepares to order a drug for a patient, let's say it's adjuvant chemotherapy for breast cancer or treating advanced breast cancer disease, two different use cases, the computer prompts them in real time in their workflow, is this patient estrogen receptor positive? If they have metastatic disease, do they have an ESR1 mutation? Do they have a mutation in PIK3CA? And all these questions, these biomarkers are, of course, coupled with an FDA-approved drug. Now, the physician may know that in real time. They may not. And, you know, they have opportunity to ask questions, not necessarily through the computer, but their colleague that's sitting next to them. But this guides the physician through best practice. Then on the data side, of course, we're trying to find these biomarkers in structured fields, and as my colleagues here will fill in the blanks, how is that data found in the unstructured field, also known as the scanned PDF document, whether it's a PET scan image. So we want to know where CAR T-cell therapy is being used. Now, we don't deliver that within US Oncology outpatient clinics. It's done in tertiary medical centers. But we might be able to search in a progress note using NLP technology. These gentlemen can talk about that better than I can. But we can take 1,000 patients with multiple myeloma diagnosed in the last year. We can find that cohort, search on the records for use cases of patient received Abecma, you know, an FDA-approved CAR T product, and, you know, where they received that, et cetera. So that's sort of a little snapshot of how we're trying to work with our data, you know, improve completeness for pharmaceutical companies, and try to reduce the day-to-day work effort of physicians in their practice.
You know, I love that general concept of removing, you know, the burden in kind of assisting the clinician, not replacing. You mentioned about data and scanned documents, which, you know, only in medicine you can take something that is digital, fax it, turn it to analog, scan it as a TIFF, and then render it unbelievably useless. Short of a carrier pigeon, it works seamlessly. I'm curious, Karim, as someone who thinks a lot about kind of how do you find those needles in the haystack? How do you actually make this data not a liability, something that we hoard and hide and protect, but something that is an asset? Tell us a little bit about Mendel.ai and kind of your journey and where are we?
So, as you've mentioned, medical records is essentially like I view it as like a DocSend or something. It's just a repo of a lot of documents and information that is essentially useless at the end of the day. It's just like a repository of the data that are very hard to use. So think of the internet without Google. It is like medical records today, essentially. It's like a big index of big data that is not indexed or searchable. So what we do at Mendel is we basically become that Google layer. We structure the unstructured data. We also take all the structured data and index it and make this data easy to interrogate. So if you want to find a patient for trial matching, if you want to do matching for NCCN guidelines, if you want to generate real-world evidence, it really all boils down to understanding the clinical data that exists through a computer rather than through a human. And some of the problems we solve is a lot of people think of it as an NLP problem, which is partially is, but it's more of a reasoning problem. Because what you see is a PDF is saying the patient is metastatic, a doctor note is saying the patient is stage 1. So now even if you have the best NLP system on earth, you have two conflicting pieces of information that cannot coexist in a longitudinal patient journey. So what we do at Mendel is we do the NLP piece, we kind of get the building blocks of the patient journey, but the more important is we have a reasoning layer that basically says a pathologist is a more trustworthy source of staging than a doctor note. But when it comes to medication, a doctor note is most probably better than like a table or something. So we're able to do that type of reasoning. We also understand that fatigue in cancer is a side effect for chemo, but in immunology, it's most probably a symptom of something. So we're able to understand the nuances of medicine as an oncologist would get it, rather than just basically index and structure the data. And what we have seen is, especially on the provider side, in the last 7 years, there's probably 20 clinical trial matching companies that came out. But we still are talking about trial matching becoming a problem. So what's the gap, right? Like there's hundreds of millions of dollars of investment that were made, many startups, many big companies, but you're not seeing that translated on the bench side. And it really boils down to reasoning. You have a lot of systems that are able to index the data, but not necessarily answer questions like a human would answer a question. Is that patient a fit for NCT, for this clinical trial or not? And that's the problem that Mendel solves. One last piece on that. We don't only understand the medical records, we also understand the NCCN guidelines. We also understand the trial, like the trial criteria is so hard to understand. It's beyond, even an oncologist sometimes would have hard times understanding the inclusion-exclusion criteria. So we're able essentially to understand clinical literature and clinical records as an oncologist would do to a certain limit, and do the matching between those and kind of generate real-world evidence or do trial matching.
No, I love the comment around it's not just an extraction or a named entity extraction, computer science. It's the reasoning aspect. A medical school note versus the oncologist note, probably a little bit different. You might want to weigh that a little bit differently. Ittai, as you think about federated learning with Rhino, kind of explain to the folks here kind of the approach that you're solving, but kind of carrying on what Karim says, about kind of it's garbage in, garbage out. How do you ensure that it's not just federated learning over data, but it's the right data, it's the right kind of curated information? How do you think about that aspect?
Yeah, it's interesting to see how this field emerged from let's digitize records, and then you do something with those digitized records, and then let's extract additional features from notes and curate data and provide the kind of like the multimodal inputs for decision making. However, one of the things that have not really been solved to date is the problem of data sharing between different institutions. And one approach to it kind of goes to throw much more money at the problem and solve some of the problem through additional data sharing that's incentivized, kind of like more and more. And another approach, which we're more leading, is reduce the friction. And one of the frictions we've seen is around the topics of sharing data between different institutions, especially when it's multimodal data, especially when it's omic data, especially when it's data that probably can't be effectively de-identified. And that ultimately leads to the fact that there's massive troves of data being untapped today that could progress medical science and medical care. Federated learning, which was actually kind of theorized by Google around 8 years ago, brings the concept of being able to train and deploy artificial intelligence algorithms without sharing data through a process of aggregating the model weights and parameters without actually sharing data itself. If you couple that together with additional privacy-enhancing technologies like confidential compute and the differential privacy, you can build a highly scalable system that would work cross organizations, solving common problems without having to share data and without having to compromise the privacy of data. That's where we come in. The idea we brought in started in 2020 when we organized a global consortium to combat COVID using data modalities that are typically pretty hard to get hold of, like outcome data from different institutions, for example, right? It was like an area where people won't just share that, and created an algorithm that proved superiority over each one of these parts and an actual synergy between medical data from across the world. We published that in Nature Medicine. There was a lot of excitement around that, and when we said, okay, how do we actually bring this into every clinic, every AMC, every HMO, every biopharma, life sciences company, and we realized that the process itself of completing an end-to-end federated computing process is actually quite complicated and requires, as you said, mitigating bias, ensuring that the data is semantically interoperable and is actually relevant to one another, supporting data persistence, supporting the provenance of data for regulatory use cases, et cetera, and that's the focus we've had with building and scaling the Rhino federated computing platform.
I love it. So I love the theme of kind of leave no data behind. You know, there's a tremendous amount of information as an asset that's probably lying untouched, unburied, that might actually liberate. And David, you commented, you know, the idea of not, you know, what's old is new, you know, repurposing our armamentarium around medicines. It's kind of, it reminds me a lot of what Ittai is talking about is, you know, leveraging all of this digital exhaust for good. I'm curious if you kind of see the correlations. Do you see your ability to unlock the next Castleman's inhibited or actually now activated by the likes of an ontology system like Mendel, or an ability to kind of create a federated learning, you know, what Rhino's doing?
Yeah, these are essential ingredients to be able to unlock the full potential of FDA-approved medicines. In the last couple of weeks, our algorithm has helped to raise up to us lidocaine, peritumoral injections of lidocaine in breast cancer patients as a potential way to improve outcomes. And you know, it seems sort of crazy. I was like, injecting lidocaine into a breast tumor before you cut it out, that improves outcomes. There was a randomized controlled trial of 1,600 patients that showed a 4% absolute risk reduction, or sorry, absolute mortality benefit, and in about a 30% relative survival benefit versus placebo. And so this is an example where there's no big lidocaine, you know, distributor out there. And so this isn't something that's going to hit headlines or really even get on a lot of our radars.
Right.
But having really strong data systems, because there's millions, maybe billions of papers, or certainly way more than that data points that are generated every year, we lose track of them. Even breast surgeons don't even know about this clinical trial of 1,600 people where if you shoot lidocaine, it helps people live longer. Like, these are the kinds of things we need really, really strong ontologies, federated systems, so that way these kinds of things can be captured, and then we can take it and make sure that patients actually benefit from it.
No, on one end, you know, if you're a clinician in the audience, you're excited, right? This is the promise. On the other end, it's daunting because, you know, every 72 days, the medical literature doubles, and our 40-watt light bulb brain doesn't. So like the reduction to practice, getting these new evidence, getting these new ideas, you know, it's not just, hey, here's a publication, here's an insight, here's the trial. You know, is that true? Is that where you're seeing kind of, you know, at the bench side or bedside rather?
Yeah, I mean, I think people would be potentially horrified if they knew how much.
Potentially horrified. Disclaimer, asterisk.
On how much of the ongoing practice of treating the cancer journey is still left at potential human fallibility. Like the beginning, you know, when you were talking about like the inconsistency in the chart and all, 80% of the upfront work for an oncologist is retrieval and processing. It's like, what did the pathology report say? And hopefully the pathologist was correct on XYZ. Let me order the molecular. What does that mean? Where were the nodes positive? That's what the stage is. Let me find out in the algorithm what's best. And then if I'm really good, what medications, you know, may matter or slant this, what polymorphisms, what pharmacogenomics. But it's all glorified retrieval at first. And then I visit, you know, the data at that point on day one to execute a plan. Now, if anyone in here wants progress or discovery or to do better in cancer, meaning new drugs or find out things that are variables that are relevant that we didn't realize were relevant, if you want that, that requires now a departure from the 25 patients you have, going back to an old molecular report somewhere, you know, under a filter, and noticing that undetermined significance now has significance last week and is going to, you know, portend a worse prognosis. And that's the reality we have. And if you're a general oncologist, in every tumor type ever. And we're just okay with that. And what distresses me is like this multi-modality approach and ability. I mean, we have a ways to go, but to be able to process on a digital path, you know, way on what's going on, the consistency of taking out human fallibility. One example I was made aware of was HER2 positivity on a blood draw, right? Circulating tumor DNA. But the breast cancer was HER2 positive in situ, so it wasn't invasive yet. And the part that was outside was not HER2 positive. Well, you get that DNA, an oncologist is going to give the HER2 therapy. It's going to fail. We don't know why it failed. You had a good chance. These are the little things that truly are relatively algorithmic and retrieval based. And I think if nothing else, it standardizes and tightens up the spillage of, again, just the volitional aspect and how much someone slept, how young their kids are, what crisis is going on, that could easily, in one decision-making, you know, step, cause a total change in trajectory of that person's well-being.
I'm going to look at, again, Bob, you know, is this kind of fair? Is this kind of how you're beginning to kind of think about this opportunity with Ontada is an ability to really kind of reduce the change management or enable the change management or the reduction of practice and help bring insights, not just show data, but bring insights to the busy practitioner?
The short answer is most definitely. I've been very encouraged. You know, the unmet need of patients is continuing to drive basically all of us here. So, and first, I just want to say a brief thank you for letting us all come up here. You know, it's been a fabulously exciting and motivating series of lectures, of talks today. So for me, who's, you know, approached the end of his
And you will get your Google hat now, Bob. Thank you.
I'm not paid by Google, not directly. I guess we all hold some stock indirectly. But, you know, I mean, back as a third-year medical student, making rounds at a community medical center where the only computers in the hospital were in the lab, and they did not have Windows. There was no Apple computing. Bill Gates wasn't drawing a salary from the work he was doing. And to retrieve the data, we were on foot, walking into the chemistry lab, the hematology lab, microbiology lab. We walked to the radiology library, pulled films, then went to the right radiology viewer, pulled the radiologist aside and says, what does this X-ray show? CAT scan. And now fast forward to the early 2000s where you pull the images up on your laptop. So it's going forward at an accelerated pace. You know, when I left practice, I would have a patient come into the office, open their last click a macro on my computer, pulled up the last progress note. I would go in and see the patient. I get that upload. I have to read the attachments because they're not structured. But then I walk into the room with the patient. I have the time. I want to encourage those here that the advances in technology already impacted the extension of my career, lessened my feeling of burnout. But the answer to the question is yes. And then to hear my younger colleagues here saying, well, it's not good enough yet at the academic medical center, or it's not good enough yet for pharma, and to be continuously dissatisfied with the status quo is what drives everything forward. So where we were 3 decades ago, where we've come to, has been extraordinary. And to hear people who identify their own treatments, I mean, it's unheard of. So kudos to my colleagues here. This is amazing.
I'm curious, just so Karim, also on, Bob talked a little bit about kind of both unstructured text, but there is this, and you did touch on it, multimodal and imaging, whether it be digital path or certainly radiology and the dreaded TIFF. Are you thinking about that ontology as well over DICOM and other kind of the entire spectrum, if you will, no pun intended, of data in building that out?
100%, and I agree with his comments, right? Like that. It's essentially a retrieval problem. It's a retrieval. And you're not trying to retrieve PDFs, or you're basically trying to retrieve a patient journey. That's actually what you're trying to retrieve. And the analogy I would give is like electric cars existed way before Tesla. But the thing that Tesla did is it made it useful to a consumer that they're able to use it, right? They understood you need a fast car. It needs to go 300 miles. It cannot be 20 miles. But the concept was there all the time. The concept of an AI sitting on top of the EMR is like the electric batteries that are not useful yet, meaning like there's no Tesla. That actually you find a physician telling you my day-to-day has changed because of this AI company. And the reason that is the case is because no one has made the AI useful to include all the EMR data and answer questions and continuously keep sifting, like HER2 now became of relevance, I need to up a note or something for the doctor. That's what we work on in Mendel. We were at ASCO this year, and they had this roundtable around AI, and one was saying they're using AI company that has raised billions of dollars and a lot of dollars that I'm not going to say their name, but they're valued at like close to $1 trillion now. And the physicians are using it, but they were saying the amount of hallucination, it's OpenAI, it doesn't matter, but it's the amount of hallucination that they have to correct makes it more time to interrogate using an AI than if they have just read the whole record on their own. And that's what I'm talking about. The idea of having a technology versus having a useful technology is a big distance, and very few companies are thinking about that distance. And unless you bridge it, you're going to have electric batteries, but you're not going to have a Tesla, essentially.
Yeah, no, I love it. I'm just curious also, Ittai, is that how you kind of think about the federated learning you're unlocking? Are you also thinking about the business model of how do you leverage the data, or do you view yourself as more of a means to liberate it for others to drive value?
So we partner with our customers and enable them to create business value through federated workloads. As part of that, we also take risk. We also are open to flexible business models. But at the end of the day, we serve a customer that has a use case. We don't develop that use case itself. Our use cases are enabling the AI stack in a federated manner.
I love it. In our last couple minutes, we'll do the speed round, because it's daunting and there is passion. I think we were talking backstage about moral injury and kind of it's frustrating, and we see all of these things before us, and it is challenging. But on the flip side, events like this of coming together, convening, is actually quite inspiring. And so for me, and maybe I'll answer my own question, is what gets me excited is just the diversity of thought, the perspectives, and the passion, and the moment that this kind of represents in total. So that's what I'm most excited about. I'm just curious, for the others, we'll forget about the pessimism, but what gets you super excited in this moment?
You probably know my answer, and that's that I think that there are a lot of drugs out there that can help a lot of people, and up until now, the tools just didn't exist to find them at scale, and now the tools do exist. The systems have been built, the ontologies are in place, the models are there, and so now I think it's just a matter of doing the work and going after the opportunities that aren't financially viable, because all of us in this room have been touched by someone with cancer. All of us have been told we've tried everything for someone that we love, and the idea that there might have actually been something there that could have saved them, I just can't imagine anything that's more important and that AI is going to be better at helping us do than using existing data on existing drugs to treat bad diseases.
For me, it's very easy. There's nothing more humbling than seeing what is incurred by a patient and their family members as well on the desperation of doing the very best for this shot. And every day when I get the papers like, well, how does this play a factor, turmeric or low glucose? And I wish I could empower or give the guidance that they're so desperately seeking with confidence. And I think that is the opportunity to really capitalize not on prospective studies, but on all the millions of very tragic cases and the observations that exist. If there's a data anchoring or some objective criteria, if we could unpack that and just have a better idea, an idea of expectation is something that I think would be a huge lift for oncologists because of that, that heartbreak.
That's awesome.
Karim?
For me, it's the idea of augmenting, think of Jarvis and Iron Man relationship. I think there's going to be doctors becoming those Iron Mans, and there's going to be the AI Jarvis. It's not going to be an AI era. It's going to be an area of augmentation between both. And as I said, bridging the gap between them. But an AI that can understand and have clinical common sense at scale, sitting next to a physician prescribing a treatment or looking at a path report is a game changer, right? It's no longer going to be how many years of experience you had as an oncologist. It's going to be how much data that the AI was trained on, and that's an easier thing to have.
Love it. Ittai?
In my case, it's taking the passion for collaboration and bringing industry stakeholders together in order to create an impactful common good play that ultimately rises, kind of like rising tide floats all boats, and delivering that value to patients and healthcare system improvement.
And I would add what is exciting me, even though I'm a clinician and somewhat of a scientist, is the redefinition of cancer biology, as has been very well reviewed throughout today's talks, going from single gene approach in our targets to multiple genes in real time, 3D spatial, looking at all the data that's available at the molecular level is very exciting. Giving the example of HIV/AIDS, having taken care of AIDS patients in this city as a medical student in the late 1980s, every patient in the hospital with an AIDS diagnosis had passed away within a year of that diagnosis. And now we talk about HIV positive, targeting 3 different protein products with effective drugs. Took 15 years in the AIDS pandemic to develop what's called highly effective antiretroviral therapy. The HIV genome is far more simplistic than the cancer genome. But I think what was described today gives me incredible enthusiasm and inspiration that we will be on the right path. How long that will take, remains to be defined.
I love it. It's a journey. So thank you for listening to our panoramic 360 view of the cyclorama of clinicians. We'll take a break.